Good evening. My name is Howard Edelman. I'm your host for Israel in the Jewish World. Our show tonight is called Asymmetry and Symmetry. Tonight's guest is Ofer Mandelboim, the Edward Crown Professor of General and Tumor Immunology at Hebrew U. He's an expert on natural killer, or NK cells, and immunological receptors. He works in the advanced field of developing personalized treatments for cancer with his deep understanding of how the immune system works on the molecular level and how NK cells can be employed to recognize and combat cancer using microRNA. If there's an early warning of danger, NK cells form large, stable, symmetrical synapses. If inhibited, asymmetrical synapses are favored. As you will learn, NK cells can read the health of other cells by recognizing the balance between activating and inhibitory agents. The theory of nuclear deterrence during the Cold War was based on a similar balance. However, in the contemporary age of irregular and asymmetrical warfare, the activating and inhibitory balances are out of whack. The stronger the party, the more inhibited in employing violence. The weaker the party, the more ready to sacrifice its young as martyrs for a cause. We have entered the age of modern asymmetrical warfare, which we will explore this evening. Stay with us. This is another in our series of discussions about innovation in the medical field in Israel, in particular with the biologists and researchers at the Institute for Medical Research, Israel, Canada. Welcome to the show.
Hi.
My guest is Ofer Mandelboim. You're a key researcher in this place. Can you briefly describe, you're doing molecular research and it has implications for sickness and health. We'll get into those, but tell me first how you came to do your research in this molecular biology field.
So I was actually born here in Jerusalem.
Yeah.
Then I did my PhD at the Weizmann Institute. And then from 95 to 99, I was at the US in Harvard in the laboratory of Jack Stromanger.
He's a very famous guy.
He is a very famous guy. This guy, actually, he's now 85 years old. He discovered the mechanism of action of penicillin, and then moved from the penicillin work to study immunology.
I see. But that's a big credit to you to be accepted into his lab, I see.
Yeah, I was very fortunate to be accepted to his lab. He's also Jewish, by the way. And then I spent 4 wonderful years. And in Jack's lab, I learned how to study NK cells, the natural killer cells, the cell that I'm working on today.
Yeah, these are NKs. Well, okay, let's describe what an NK cell is, this natural killer. By the way, we should say that when I was first in, I was told it's Killer Mandelboim. I thought, this is a tough guy. And I learned there's a paratrooper. It's a really tough guy, but that isn't why you were called Killer.
Right. So these cells, actually, we'll see later on that these cells are not only killer cells, they also help pregnancy. But they were discovered in the 70s, actually by Swedish scientists. And he noticed that these cells could kill tumor cells and virus-infected cells without any activation. So that's why he named them natural killer cells. So they can naturally kill tumor cells and viruses. And back then, people didn't know how to identify them, how to work with them, and today we know quite a lot about these cells.
They're very different, they operate very differently in doing this killing of viruses, etc., than, say, when you get a macrophage and they seem to kill the cell even, don't they?
Right.
It's not just the invader.
Right. So actually, they are one of the first line of defense. So when people
get the infantry.
Exactly. So people, but they are kind of maybe more sophisticated. So when we are being attacked by a pathogen, for example, so they start to operate so the first cells to actually tackle maybe the simple infantry are the macrophages.
Right.
They stay a bit behind the NK cells and wait about 24 hours, and then they move to the site of entry. And then they can recognize specifically tumor cells, virus-infected cells, bacteria and parasites, and then they will kill these invaders to protect us from, I don't know, these first enemies.
So, but this second layer, they essentially, exude some kind of poison that doesn't kill the rest of the tissue, just the tissue directly involved.
Right, so that's one of the things that we are working on. So they have a special set. So how could they discriminate between the self and non-self, between enemy and regular healthy cells? They have special receptors that are programmed to recognize many different pathogens. So they move to so they've been attracted to the site of injury within 24 hours. Then they recognize specifically the cells which are infected, let's say, with the virus, and can kill these infected cells in a direct hit. So they bind to the cell and recognize the foreign cells with their receptors, with their proteins, and then deliver the little hit and kill the cell.
It's heat that kills it.
It hit. No, the little hit.
Hit. Oh, I thought it's your Hebrew English accent. So what? But they hit them with what? A protein?
Yeah. So they have. So they kind of form a synapse. So they recognize. So you have two. You have the NK cells and the invader. They recognize the invader via special receptors, and they deliver cytotoxic granules into this towards the infected cells, for example, and simply kill it within 10 minutes. The cell is completely dead.
You know, when I read this stuff, because I write on just war theory, so I think of this. Does this have any application by some, analogy to just war theory? Because they just wipe everything out.
Right.
They wipe it clean, as I understand from reading the.
But it has specificity, so they will recognize only infected cells or tumor cells. They will not kill normal cells, otherwise we will be facing a problem of autoimmunity. Right. In fact, we have recently found out that they are kind of important even during type 1 diabetes. So during diabetes development, they also come in contact with the beta cells, we don't know why, of the pancreas, and just start to kill them.
Really?
And they contribute to diabetes development, yeah.
So I see.
So when something gets wrong, they can be really dangerous. They can kill within a few minutes.
Our own parts of our body start killing the...
Exactly.
So the lymph... Oh, I didn't know that at all. Yeah. I remember my first lecture when I was in medical school a great lecturer came in for Chicago and taught us about... Because diabetes is a big thing in Toronto because of Banting and Best. So he taught us how it works and the insulin, etc. And after all these years, nobody knows how it works.
Exactly.
And yet he taught, how much they did and how it worked. And
so we just discovered that the NK cells really comes there as well. I mean, together with the T cells and that are killing the beta cells. And so if we will be able to try to find ways to block the action of NK cells, maybe we can help diabetics.
I see. But your big breakthrough paper came through. You have had a meteoric career since you've left Harvard. Maybe you had one at Harvard, I don't know. But when I read the papers, everybody cites this 2006 paper in which you radically changed the paradigm of NK cells. Because you read all the papers, and I'm a total amateur, and they seem they're just killers.
Right.
And you discover they're not just killers. They do something quite creative.
Right. Actually, this work was inspired by a Canadian researcher by the name of Anne Croy.
Anne Croy?
Yes. So actually,
where does she work?
She now, I think she used to be in Toronto, but I think she moved now.
I see.
So actually, she discovered about 20 or more years ago that in the mouse. So there is a really amazing question. How come pregnancy survives? So the fetus, half of the genetic code is coming from the father.
Oh, so it's a foreign substance.
Exactly, it's a foreign substance.
So how does the woman tolerate it?
Exactly, how does the woman tolerate it? So there are many answers to this, but maybe one of the answers is that in the point of contact between mom and baby, there are no immune cells. But this is not the case. In the point of contact between mom and baby, there are immune cells, but the majority of these cells are it's completely amazing, are natural killer cells. And no one really knew what they are doing there.
When was it discovered that the majority
So this
were
was
natural?
discovered maybe 30 years ago by a woman from Cambridge, Ashley Moffat. So she kind of noticed that in the point of contact between mom and baby, there are large cells, which are anchor cells, and no one really knows what they are doing there. Then Anne showed in the mouse that maybe these anchor cells help the fetus to grow. But pregnancy in mouse is completely different than humans, and because of that, and also the research in humans is quite difficult. So because of that, until we, found out, I think we published our paper, no one really know what's going on in humans. So
we don't want to get into your paper yet, because
Okay.
you have to take a break.
Okay.
We'll hold everybody in suspense on this
Okay.
great revelation that
Okay.
comes from your research and how you came about
Okay.
it. Okay, so we'll take the break now.
Okay.